Hi, this is Shankar Reddy, PhD Plant Pathology. So I think so far the videos what we have published is really useful for you guys in the on the way of preparing NET 2023, not only for NET but also for SMS and STO. So this is just like like last time, last minute preparation or one final recap to all the videos that what we have published. So I think many of you guys are writing for the first time, right? So don't worry about exams, how it will is going to be. I think you guys having a little bit tension, or maybe what can I say, that excitement, right? So I will share you few of my experiences because I'm going to write my fifth time. So this is for just for experience. If I want to share experience, at least I need to have some knowledge, right? So this is my fifth time. So I will share few of my experiences that what I gone through on the last 4 nights. So I started writing in 2018 and 2018 1, 2018 2, 2019 and 2021. So I cleared my net in 2018 itself, and so we'll see what are all the topics we can expect. But don't think that the questions will exactly come from this the same section only, because the question paper is going to take by someone else, not by me, right? Not by me or not by you or someone else, so that we can tell exact the questions will come from this topic or that topic. So I think like most experienced people and one of the prestigious people will take the question paper. So I think the question paper will be, I expect this time going to be okay, because the last 2 times the question paper was a little bit tricky. I can't say difficult, but I would say a little bit tricky, like something depthy observations and depth understanding will be required. So the exam was just like announced just 45 or 50 days before, and the exam is approaching very fast. I think very few days, couple of days left, right? So I will share what are all the few other topics that we can expect. So the first thing is from general plant pathology. So from general plant pathology, what we can expect is like fungal cell wall is made up of, cell wall compositions, or fungal cell wall is made up of, or bacterial cell wall is made up of, or what is the oldest plant pathological society, or like Western blotting techniques. So I think that will comes under not laboratory techniques and equipment that we will discuss there. And along that we can expect like phytopathological societies, what are the phytopathological society there, and when the Indian Phytopathology Society was established. Such kind of questions we may expect from general plant pathology. In general, we may expect from various other topics too. So but when it comes to the history, see, with the experience that what I have so already wrote 4 times, so at least 5 to 10 questions, I would say 7 to 10 questions will come from history. So mostly general questions and direct questions only will be asked, like the most important contributions like Anton de Barry, P.A. Michelli, C.H. Parsoon, E.M. Fries, and Tulsan brothers, and some other direct topics only we can expect, and the books that they published. For example, I will tell you Nova Plantarum Genera, that was written by P.A. Michelli in 1729. So he is considered as father of mycology. But father of plant pathology is Anton de Barry. So Anton de Barry had lots of contributions when it comes to plant pathology. That's why we called him as father of plant pathology. So like books which was written by Nova Plantarum Genera, fungi and plant diseases, or fungi and diseases in plants was written by, and such kind of books are there, like author names who wrote that particular books. Sometimes we may also expect years. So that is also will happen definitely. I think the histories will be easily available in various sources, and we also published a video regarding history, not the video that we published recently, not like multiple choice or MCQs. I think we already published the video regarding history in almost 1.5 years, 2 years before. I think you guys those who didn't have not seen, you just go to playlist and principles or introduction to plant pathology. The first video is history of plant pathology. I think we have talked around 1 hour especially about history only. I think that history is more than enough. I may expect more than enough. The mycology or bacteriology, and like Robert Koch, E.F. Smith, he is considered as the father of phytobacteriology or plant bacteriology because of the contributions. and T.J. Burrill, he's the one who discovered the first plant pathogenic bacterium, that is Irvinia fire blight of apple, Irvinia amylovora. Before it is called as Micrococcus amylovorus, not amylovora. This question was actually asked. Micrococcus amylovora. What is the first name of first plant bacterial? There is fire blight of apple. So, and their transmissions also we can expect, like virology, like we have Ivanovsky, MJ Bijernek, and MJ Bijernek, Ivanovsky, and one more WM Stanley, Bowden and Pyre, Geirer and Scram, and one more Adolf Meyer. So such kind of contributions we may expect from history section. So when it comes to epidemiology, Van der Planck or some other like Nagarajan and Singh and the books which was written by, I told you, you just concentrate the books which was written by the particular scientists, especially famous scientists or famous botanists we can say. The books which were written by their major contributions. I'll tell you one more thing, like genus Allomyces was created by E.J. Butler. So such kind of questions we may expect. And books which was written by E.J. Butler, or what is the first book that is completely devoted to plant pathology, because so many books was written before, but disease of crop, cultivated crops and their causes and their control is the first book that is completely related to plant pathology, that was written by J.G. Kuhn in 1858. Year is also important sometimes. So we may expect such kind of questions. So that is from history section. I don't want to take your time more. And when it comes to mycology section, it's a very broad spectrum of area, so it will be very difficult so that where we can expect. But I will tell you some common topics, like in women's, if you take Pythium phytophthora and downy mildews. So sporangial spore arrangement of downy mildews, Pythium phytophthora, what are the different kinds of spores produced by. For example, one more thing I would like to tell you is what are the sexual and asexual spores produced by fungi. For example, if you take oospore and zygospores are the diploid resting spores. When it comes to ascospores and basidiospores are the haploid non-resting spores. So such kind of questions also we may expect, like then also ask, we may also expect like what is the special feature of Pythium. So Pythium won't produce any Hastoria and Aprosoria, and you know, like what is the special feature of Pythium? The formation of vesicles above the sporangia is the special feature, because when the protoplasmic material was there in the sporangia, it will remains as a protoplasmic material. When the protoplasmic material when shifted or migrated to vesicle, where the compartmentization, we would say juice pore formation. So such kind of questions we may expect, like vesicle is the formation of vesicle is the special feature of Pythium. And the questions like what is the special feature of Pythium? One more special feature is it is a biflagellate with anterior tinsel and posterior whiplash. Whiplash means no hair. Tinsel means with hair. So such kind of questions we may expect. And the cell wall compositions, like fungal cell wall is made up of chitin, or bacterial cell wall is made up of mucopeptides or peptidoglycan, or you know, Womycota group of cell wall is made up of like it is made up of cellulose. But few fungi, even though it comes under Womycota group of fungi, their cell wall is made up of chitin. For example, I will tell you, like chitin. Chitin containing Womycetes group of fungi is Acaila, Aphodogyla, Saprolignia, Leptometus. So such kind of fungi, even though they comes under Womycota group of fungi, their cell wall is made up of cellulose. So I'm going little fast because since most of the guys are MSc, so that I hope it will understand. If it is a UG level, I may deal in a separate way. So hope you guys understand and catch the speed. And when it comes to other sections like Zygomycota, in only two fungi are there, like Rhizopus and Mucor. Maybe stolons is a special feature, or maybe the formation of stolons or something like that. They may ask. Aprosorium is the attachment organ, and Hastoria is the nutrient absorbing organ. So they can also ask what are the shapes of Hastoria. For example, I will tell you one example. So finger-shaped Hastoria is produced by, actually finger-shaped Hastoria is produced by Phytophthora, and few other species also produce finger-shaped Hastoria. So such kind of questions we may expect, like powdery mildews or types of powdery mildews, Idium, Idiopsis, Ovuloropsis type of there, and types of Clistothesia, Sparotheca, dichotomously branched or bulbous appendages or coiled tips, something such kind of things will be there, like the Clistothesia with the special appendages. So such kind of questions we may expect. And you know, like when it comes to Fusarium, Microconidia, and the Chlamydospores it will produce. So, and They can also ask what are the fungi which produce sclerotium as a resting spore. Sclerotium ralphsi, clariceps, microfomina, saraclodium, rhizoctoni. There are all the fungi which produce sclerotium as a resting spores, right? So such kind of questions we may expect, and other things like from mycorrhiza side, ascomycota is a very big thing. Mushrooms also we can expect a few questions. I think I already published the videos in a very lucrative manner, or very the important points I concentrated, and we delivered our level best. Hope it will be useful. And when it comes to bacteriology section, like gram positive and gram negative bacteria, like mucopeptide compositions, or what will be the color of gram positive and gram negative bacteria at the end of the staining, and what are the different kinds of toxins that is produced by plant pathogenic bacteria, and something like that they can ask. And from bacteriology section, we can also expect like bacterial growth curves. What are the different kinds of bacterial growth curves? Along with that, I would say other things like toxins and genome structures, like what is the first plant pathogenic fungi to be completely genome sequenced, what is the first plant pathogenic bacteria to be completely genome sequenced. Such kind of questions we may expect. From virology, we can see like history, and along with that sizes of plant viruses, and coconut kadang, what is the size and length of coconut kadang virus, especially vectors. So virus is vectors are very, very important. We can definitely expect few questions from vectors. And epidemiology and physiological plant pathology. See, for physiological and molecular plant pathology, it will be also like a it's a puzzling topic for me, because I really tried a lot, but few topics I really can't able to connect because of this depthy molecular and physiological side. So I was not that much well versed about physiological and molecular plant pathology, but we have published some videos like phytotoxins, or different kinds of PR proteins, or different kinds of toxins produced by this, and along with that theories, hypothesis, some kind of things will be there. ETL, PTL, ETI, PTI, and systemic acquired resistance, induced systemic resistance. Such kind of videos we already published, and hope it will be useful. And when it comes to applied plant pathology, that is chemicals and plant disease management, like trade names, the specific fungicide. For example, I will tell you one thing: rice blast is managed by the specific fungicide that can be recommended for rice blast is tricyclazole. And we can also like edipinphos and ibuprofen also there. Trade names and the specific fungicide, sometimes resistant varieties also, especially vectors also we can concentrate here. Actually will comes under virology section. That one, and along with we can expect the chemicals are the group of fungicides, mode of action. Yeah, I forgot to tell you, the mode of action is also very, very important. I think in previous question papers also I have seen asking mode of actions, at least one or two mode of actions, like basic questions. So they're like new generation fungicides also, they will ask like mode of action of strobilurin group of fungicides, where actually the strobilurin group of fungicides are isolated from Strobilurus tenax, let's say mushroom, right? So such kind of questions we may expect. And mushroom sections, so the first first mushroom known to humans, or what are the cultivated species of mushroom, and their scientific names. I would say botanical names. So scientific names, and maybe varieties, sometimes maybe varieties. Agar-agar is produced from. That's a quite common question. If you see from 2006, so agar-agar is produced from. That is a very standard and quite common question. And bacteria, in bacteria, FVB, like Fastidious vascular bacteria also there, xylem limited, phloem limited, such kind of questions. And pathogenic names also will be there. There is a separate question, separate section in syllabus is there. That is the disease of crops. Don't think that whatever we have studied, we may expect the same kind of questions, like the same kind of pathogens. So like periwinkle yellows caused by. So different kinds of questions we may ask. So apple proliferation is a phytoplasmal disease actually. So such kind of different kinds of questions and different kinds of plant pathogens, there might be a chance. And we may expect important questions also. But I will tell you one thing. So for doing, while we are studying UG and PG, or even in PhDs, mostly we have, for example, if you see rice, there are more than 120 diseases are there in rice, but what we are studying is just 15 or 10 to 15, right? So, but the remaining is also, we can't say that those are not important, because the syllabus is already given very clear. So they can ask questions wherever they want. So we can't go and ask the questions where it is out of syllabus or it is out of something like that. So because everything is clearly given. So we are not kids anymore, right? So we need to explore more. So second thing is that is about applied plant pathology and physiological plant pathology. So when it comes to laboratory equipments, right? Laboratory equipments, like who discovered the particular equipment, or what are the functions behind, or the temperature. For example, if you see autoclave, so there are different kinds of sets of temperatures are there in autoclave. Not only autoclave, but also if you see hot air oven. Actually, hot air oven was discovered by Louis Pasteur. Laminar flow chamber was discovered by Whitefield. He's also called as Mr. Clean. And autoclave was discovered by Charles Chamberlain. So the discoveries, the principles behind the temperature, how much time we need to maintain the temperature. So such kind of questions also we may expect. And along with their isolation techniques, what are the surface disinfectant agents or chemicals, long-term storages, and so various kinds of things are already there. And along with that, we may also expect advanced techniques and technologies like PCR, ELISA, and PacBios, like next generation sequencing, northern blotting, southern blotting. What is northern blotting used for? What is southern blotting used for? What are all the things? So the time is very less, so that we did our level best. So if we have lots of time, we can also take topic wise videos, and I also will figure it out the expected topics, or what are the questions we can expect, something like that videos we can do. So we are also planning to start a couple of new series of videos, especially for mycology and especially for the net, especially to the students who have no idea about it, where to start, how to start. So such kind of students, we are also having a future idea of how to start and all those things. Where to start and how to study. So such kind of things. So we are planning to start a separate lecture videos, not only for that but also for mycology, bacteriology, and various other topics. So if you guys will follow us, I never ask you to follow us. It's your wish, whether you want to follow or not, that's your wish. So, but if you follow, you can get a complete ideology about plant pathology. So if you want more and more knowledge, or if you want more information about many more topics and all those things, I would suggest one of my book. I can't say you please go and buy. I would suggest this book. If you are interested, you can buy this book, because it is like if you study this, A Vision into Plant Pathology, a complete student version. So this, trust me, this will be a complete student version for the students who are in the initial levels or and the moderate levels. I would say that encyclopedia for students, not for everyone. You please make sure of it. Encyclopedia for students. And if you see the volume, this is volume 1 and this is volume 2. So many things. So I think there are plenty of books are available in market, but the specialty of this book is, so while I was studying, or while I was there in the initial stages of plant pathology, or initial stages of learning, I've seen so many books, but there is no book that covers the complete topics of each and everything. Starts with mycology, bacteriology, virology. If one thing will be there, other thing won't be there. Like mushroom, if you see nematology won't be there in this kind of books. So we need to separate such separately. Applied plant pathology also, it will be most difficult to find in the same book. Along with that, nematology, applied plant pathology, so many things are there. And phanerogomic plant parasites also. I forgot to tell you that phanerogomic plant parasites, nutritional disorders also, we can definitely expect a very good amount of questions. Like phanerogomic plant parasite, we may expect 2 to 3 questions. There are only 4 are there, right? Cuscuta, Loranthus, Verbanchis, Striga. The same questions only, they will shuffle it and they will ask in a very confusing way. So on nutritional disorders, there's some couple of standard questions are there. Little leaf of brinjal is caused by, rice cera is caused by, and so such kind of some standard questions are there. So you guys just please go through, and videos that what we have published is already uploaded to our website www.geekyresearcher.com. Hope you guys will go and download the videos. And for further more information you guys want, if you want, you can buy this book, and this book will be available in Kalyani Publishers official website. And you guys also contact us for further more details or further more guidance. We do absolutely do free of cost, because we do serve students, because it's not like paying something, or teaching something not like that. This book will be definitely useful. Hope interested guys can take a look into, and this will be available in Kalyani Publishers website. And I won't say much about this book, because if I talk much more about this book, you guys will thought that he wrote the book, that's why he's talking about. So this is just 2 books only. I'm writing a couple of other books also. Trust me, this will be like a complete series For each and every single student, this will be like a complete the books that I am going to publish, it's like a complete series. You no need to look into some other books or any kind of books. Anyway, I don't want to take much more time. So those who are writing or who already experience, I think most of the guys who are writing are like maybe freshers or maybe having experience, 1 or 2 years experience. So we should very all the best to each and everyone who are writing NET 2023 SMS and STO exams. And one more thing I would like to tell is ever tried, ever failed, try again and fail again. But remember, every time when you are failing, fail better every time. Okay, so I know people who are still writing their NET exam for 8 times or 9 times, still they couldn't able to manage to get pass marks. So don't worry about results and all those things. Do your level best. Hope everyone will pass this exam, and once after getting the results, we will definitely meet once again. And wish you very all the best to each and everyone. Stay geeky and stay tuned. We are Team Geeky Researchers.